15 slides total.
Okay, no problem, no problem. We'll see how many we can do today, and then again tomorrow we have class.
Yes, sir.
So accordingly.
Good morning, sir.
Good morning, Sakshi. We will just wait two others to join, then we will start.
Yes, sir.
Good morning, sir.
Good morning, Nehal.
सर अल्फाबेटिक वाइज हम पीपीटी देंगे सर, मतलब पहले निशा.
It's perfectly fine. It's very good.
ओके सर.
It's very good.
एक्सक्यूज मी सर.
Please, Sakshi.
सर जो एसआरएम यूनिवर्सिटी की जो बस है सर, वो सोनीपत की किस रूट से आती है? मीन्स हमारे यहाँ से.
आपके यहाँ से सोनीपत होके तो आती है.
यस सर, सोनीपत से सर. मीन्स हाँ, किधर से आती है सर? आईटीआई चौक के पास से सर.
आपकी आवाज कट रही है, साक्षी.
सर सोनीपत से किस साइड से आती है सर?
सोनीपत से आप एक्चुअली मैं उस बस में जो कोई आता है उसका मैं नंबर आपको भेज देता हूँ, आप बात कर लेना.
यस सर, थैंक यू सर.
वेलकम. आई एम जस्ट सेंडिंग यू नाउ. साक्षी मैंने आपको भेज दिया, आप बात कर लेना.
ओके सर.
शिखर, गुड मॉर्निंग.
Sir, good morning.
हाउ आर यू शिखर?
Sir, fine.
वेरी गुड, वेरी गुड. अनिशा,
Yes, sir.
आर यू रेडी फॉर प्रेजेंटेशन?
Yes, sir.
Then now is your time. You can start.
Yes, sir. Is it visible, sir?
It's visible.
Okay. Good
And
morning to one and all present.
yeah.
Yes, sir.
Good morning. Please start.
So my topic for today's presentation is antibiotics, that is penicillin and streptomycin and single cell protein, basically industrial production of all these. So let's begin. The first one is penicillin. Before coming on to the production process of penicillin, I would like to brief about it quickly. Penicillin was the first true naturally occurring antibiotic ever discovered. So it was the great medical breakthrough 2, and these are a group of beta-lactam containing bactericidal antibiotics. Bactericidal means that it prevents the growth of bacteria. If we look into its history, that from when it was discovered, then in 1928, Scottish biologist Alexander Fleming discovered that the Staphylococcus culture, which he had mistakenly left growing, was contaminated with a mold which had destroyed the bacteria. As you can see here in this picture, this was the Staphylococcus culture which was contaminated, and after testing it, he found that it was Penicillium notatum, and later on penicillin was purified from it. So the first time penicillin was obtained was from Penicillium notatum, and now today penicillin is produced on a large scale to treat bacterial infections. Talking about its structure, it have a beta-lactam functional group, thus belongs to the beta-lactam antibiotic group. They all have basic ring-like structure derived from two amino acids, that is valine and cysteine, via a tripeptide intermediate. And this third amino acid of this tripeptide is replaced by an acyl group. The nature of this acyl group produces specific properties on different types of penicillin. Now let's see how this penicillin is produced industrially. So this can be broadly classified into two processes. The first is upstream processing, which is a process or technology that leads to the synthesis of a product. It includes the exploration, development, and production. In this process, the medium is designed to provide the organism with all the nutrients that it requires to grow. Now the second process is downstream processing. This process includes the extraction and purification of a biotechnological product from fermentation. There are several stages in downstream processing. First of which is removal of cells. That is the first step in product recovery. Is the separation of whole cells and other insoluble ingredients from culture broth by either filtration or centrifugation. Then the second stage is raw materials, for which carbon sources and nitrogen sources are generally used. Then mineral sources are also required, for which calcium can be added. This figure is an outline of the flow chart for the industrial production of penicillin. In as here the lyophilized culture of spores. Lyophilized means it is a process by which water is removed from a product, or you can say it is a kind of frozen culture. So the lyophilized culture of spores is cultivated for inoculum development, which is then transferred to a pre-fermenter and then to fermenter, in which the essential nutrients required by the organism are supplied. Then comes the product recovery stage, as I told you earlier. In this, it is filtered, then placed in a cooling tank, from where the further purification process is carried out. Now, this figure is illustrating the penicillin production in some more detail. As penicillin production is an aerobic process, so therefore continuous supply of oxygen is required to the growing culture. And the pH is maintained around 6.5, and optimal temperature is in the range of 25 to 27 degrees Celsius. This production is usually carried out by submerged process. The medium used for fermentation consists of corn steep liquor, that is sugar, and carbon source, usually lactose. An addition of yeast extract is done for a good supply of nitrogen. The talking about the starting culture, as I told you already, that earlier penicillium notatum was used for the large scale production of penicillins. So these are added. Currently, the Penicillium chrysogenum are preferred. After adding medium and starter culture, the fresh medium is added 10 times in 6 days. After that, for the filtration process, there is a rotating filter which will separate the product. Some of it can be used as animal feed. And for the recovery of penicillin, as the fermentation is complete, the broth containing about 1% is processed for extraction. Now penicillin is recovered by solvent, that is butyl acetate. Then potassium or sodium acetate is added to remove the impurities. The yield of penicillin is around 90% at this stage, which can further be treated chemically or enzymatically to make new antibiotics. So this was all about the production of penicillin. Now the next one is streptomycin. It is also an bactericidal antibiotic produced from Streptomyces griseus and is effective against gram-negative bacteria and tuberculosis bacterium, that is Mycobacterium tuberculosis, and it is also proved to be useful in the treatment of infections caused by gram-positive, especially those which are resistant to penicillin. It was developed by Scats, Boogie and Waxman in 1944. Talking about its chemical structure, then streptomycin is a aminoglycoside antibiotic, and the basic compound which is available as hydrochloride HCl as a crystalline hydrochloride double salts with calcium chloride or a phosphate and dihydrostreptomycin. Coming on to the production of streptomycin, there are phases during it. Mainly three phases are there. The first phase, it takes about 24 hours to 48 hours, and it is characterized by the rapid growth and formation of abundant mycelium occurs during this phase. The pH rises to 8 due to the release of ammonia into the medium, and due to the proteolytic activity of Streptomyces griseus, glucose is utilized slowly and little production of streptomycin is witnessed. Now in the second phase, it lasts for 2 days. Streptomycin production takes place at a rapid rate without increase in the mycelial growth. The ammonium released in the first phase is utilized here, which results in the decrease of pH to 7.6, and the glucose and oxygen are required in large quantity during this phase. Coming on to the third phase, in this phase, cell undergoes lysis, releasing ammonia and increase in the pH, which falls again after a period of continuous streptomycin production. The requirement of oxygen decreases and the content of the medium, including sugar, gets exhausted. Finally, the streptomycin production ceases. Approximately a yield of approximately 1200 micrograms per milliliter of streptomycin is obtained this way. The production of streptomycin can be better understood by the help of this figure. Firstly, spores of Streptomyces griseus is maintained lyophilized in a carrier, such as sterile skimmed milk is employed as a stock culture. Now, these spores from the stock culture are then transferred to a sporulation medium to provide enough sporulated growth to initiate liquid culture. Buildup of mycelium inoculum in a flask, generally a slant, is prepared. After sufficient mycelial growth, it is fed to production fermenter, where a fermentation is completed. By using filtration technique, mycelium is segregated from the broth. In one procedure, the streptomycin is absorbed from the broth into the activated carbon and hence clubbed from the carbon with dilute. Then it passes through column containing cation exchange resins to absorb streptomycin from the broth. Column is washed with water. Now this streptomycin is eluted with HCl before concentration rises into the absorbed streptomycin from vacuum. In addition of acetone to the filtrate for the precipitation of the antibiotic, finally precipitate of antibiotics is washed with acetone and then dried. Then it is mixed in methanol for precipitation as the pure streptomycin calcium chloride complex. So the streptomycin, we have seen the industrial production of streptomycin, then why it is important. So the uses of streptomycin is that it is effective against tuberculosis and infective endocarditis, which is caused by enterococcus when the organism is not sensitive to gentamicin. It is also effective against tularemia infection, which is caused by the bacterium Francisella tularensis. It is used to treat plague caused by Yersinia pestis that has historically been treated with it as the first line treatment. So this is all about streptomycin production. Now the last topic is single cell protein production. What is single cell protein? single cell protein, that is SCP, refers to the microbial cells or total protein extracted from pure microbial cell culture, which can then be used as protein supplement for humans or animals. Besides its high protein content of about 60 to 80% of dry cell weight, SCP also contain fats, carbohydrates, nucleic acid, vitamins, and minerals. And there are a variety of microorganisms which can be used for the production of single cell protein, such as fungi, example Aspergillus niger, yeast, for example Saccharomyces cerevisiae, and the algae, example spirulina, or bacteria such as lactobacillus. So the selection of microorganisms depends upon the composition of proteins, fats, nucleic acid. And if we look up to the protein content of fungi, it is approximately 30 to 40%. In algae, it is 40 to 60%. In yeast, it is 45 to 55%, and in bacteria, it is about 50 to 65%. Likewise, other composition are also listed here in this table. Now, this SCP is suitable for human consumption. It is also considered as food grade. It is regarded as feed grade when it is used. And there are many advantages of using microorganisms for single cell production. The first is that microorganisms grow at very rapid rate under providing optimal culture condition, and some microbes double their mass in less than 30 minutes. And the second advantage being using microorganisms for the production of single cell protein is that also the quality and quantity of a protein content in a microorganism is better when compared to higher plants and animals. And one plus point with microorganisms is that they can be easily handled and subjected to various genetic manipulation in order to improve the final product. So if we talk about the production of single cell protein, then it is carried out in the following steps. The very first step is selection of suitable strain. It is very critical step as the quality of protein depends totally on the microbe that is used for the production. Thus, careful selection of the strain should be done. Like it should be determined that the selected strain should not produce any toxin or undesirable effect in the consumer. Now, the criteria for the selection of microorganism can be that it should be fast growing microorganism and for massive output, and sometimes the pH of the growth medium also changes during the process of fermentation. So it is the activity of microbe to tolerate a wide range of pH, and thus it impacts the overall production of single cell protein. The second step for the industrial production of single cell protein substrate for single cell protein production. So the nature of the raw materials that are supplying substrate is very crucial for SCP production. Substrates are selected on the basis of their abundance in the production area. Like carbohydrate derived sources are widely used for this purpose. The reason being microbial dependence on the monosaccharide and disaccharide for the multiplication, which serves as building block of substrate. Also, carbohydrates are easily available and widely distributed in nature and have high energy value. And the third step is fermentation. Fermentation is the main process involved in the production of SCP, which are of two types, that is submerged fermentation and semi-solid fermentation. I will tell you about these two in the next slide. So whatever the steps we discussed so far are shown here in this flowchart representation. As we now know that the raw materials, the nature of the raw materials supplying substrate is crucial for single cell protein production. The most commonly used raw materials may be grouped in the following categories, like waste product, agricultural sources, high energy sources, and carbon dioxide. सर नहीं है क्लास में? Hello, am I audible?
Yes, Anisha, you are audible.
सर ने left का meeting?
सर कहाँ गए? लगता है
सर तो है ही नहीं, एक मिनट तो
सर
तो
तो सुन
मैं
ही नहीं रहे।
कब से बोल रही हूँ. मतलब सर ने सुनी नहीं मेरी प्रेजेंटेशन?
नहीं सर ने शायद अभी लेफ्ट किया है, मेरा भी ध्यान नहीं था कि सर ने मतलब पता नहीं कब चले गए, बट सर अभी तो थे 2 मिनट पहले।
अब क्या करूं?
नहीं नेहल अभी कहाँ।
सर थे तो अभी ऊपर आ रहा था। अरे कोई नहीं अनीशा, तुम दोबारा बोल देना। ये ले,
ऊपर से
मैं इतनी सारी वापस नहीं बोल सकती यार।
ठीक है मत बोलना, ऐसा करेंगे, मेरे को खत्म करनी है आज, मैं इतनी टेंशन
ठीक
लूंगी
है।
आज। मैं बोल दूंगी हर सर हमने समझ ली, अनीशा ने और नेहल ने भी स्टार्ट कर दी थी नेक्स्ट, मेरी भी हो गई।
सर को मैसेज
अब
करूं क्या मैं?
सर आ रहे हैं, बोल दिया, मैसेज कर दिया सर बोल।
अच्छा, तो फिर सर आएंगे तभी मैं कंटिन्यू करूं?
नहीं मैं बोल दूंगी सर को, अनीशा की हो गई सर, नेहल की भी हो गई, अब बस साक्षी बची है, साक्षी की ले लो।
तो फिर मैं अभी कंटिन्यू करूं? सर के आने से पहले 1 मिनट रुक जाते, सर आ जाए तो। बस
सर
होने वाली है फिर।
साक्षी, साक्षी तुझे देनी है क्या नेक्स्ट? इसके बाद मैं ही दूंगी। क्या हो गया?
Hello, Anisha.
यस सर।
Yeah, sorry, I lost the net, electricity, then network. I come back. So, Anisha, can you just
यस
go
सर।
to the back slide, the single cell protein that?
सर दिस स्लाइड।
Yes, yeah,
ओके सर।
because here is the problem. It's from the Baiju, right? Can you see in the top?
यस सर, यस सर।
So it should not be like this. You need to remove it,
ओके सर।
or you need to give the references.
यस सर।
Okay, you can start from here.
Okay, so the single cell protein refers to the microbial cells or total protein which are extracted from pure microbial cell culture, which are then used as protein supplement for humans or animals. Besides high protein content, SCP also contain fat, carbohydrate, nucleic acid, vitamins, and minerals. And there are a wide variety of microorganisms which can be used for the production of single cell protein, such as fungi. Example of fungi include Aspergillus niger, yeast such as Saccharomyces cerevisiae, algae such as spirulina, or bacteria such as lactobacillus. So the selection of microorganisms depends upon the composition of proteins, fats, nucleic acid, and if we look up to the protein content of fungi, it is approximately 30 to 40%. In algae, it is 40 to 60%. In yeast, it is 45 to 55%, and in bacteria, it is 50 to 65%. Likewise, other compositions are also listed here in this table. SCP is suitable for human consumption, and it is also considered as a food grade. Now, there are many advantages of using microorganisms for single cell production. The first is that microorganisms grow at a very rapid rate under optimal culture condition, and some microbes double than mass in less than 30 minutes. And the second point being also the quality and quantity of a protein content in microorganisms is better as compared to higher plants and animals. And one plus point with microorganisms is that they can be easily handled and subjected to genetic manipulation in order to improve the final product. So if we talk about the production of single cell protein, then it is carried out in the following steps. The first step is selection of suitable strain. It is a very critical step as the quality of protein depends totally on the microbe that is used for the production. Thus, careful selection of the strain should be done. Like it should be determined that the selected strain should not produce any toxin or undesirable effect in the consumer. Now, the criteria for selection of microorganism can be that it should be fast growing microorganism, also required for getting massive output, and sometimes the pH of the growth medium also change during the process of fermentation. So it is the ability of microbes to tolerate a wide range of pH, and thus it impacts the overall production of single cell protein. The second step is substrate for single cell protein production. The nature of the raw materials supplying substrate is very crucial for single cell protein. Substrates are selected on the basis of their abundance in the production area, like carbohydrates. Derived sources are widely used for this purpose. The reason being microbial dependence on the monosaccharides and disaccharides for their multiplication, which serves as building block of substrate. Also, carbohydrates are easily available and widely distributed in nature and have high energy value. The third step is fermentation, which can be carried out in two two types: the submerged fermentation and semi-solid fermentation. And I will tell you about these two in my next slide. So whatever the steps we discussed so far are shown here in flowchart. As we now know that the nature of the raw material supplying substrate is crucial for the single cell protein. So the most commonly used raw materials may be grouped in the following categories, like waste products, agricultural sources, high energy value, and carbon dioxide. For the production of single cell protein from waste, there are several materials that have no useful purpose and they are collectively called as waste. Example: molasses, whey, animal manure, sewage, uh, date waste, etc. These waste products are produced in various industries and other biological processes, largely contributing to the environmental pollution. But these can be utilized for the production of SCP, like conversion of low cost organic waste to useful product. But there has been very limited process for the large scale production of SCP from waste, mainly because of transportation cost or technical difficulties. Now the next one is production of SCP from agricultural sources. The natural waste wood sources containing cellulose, hemicellulose, and lignin are used for the production of single cell protein. It is essential to break down these cellulosic compounds into fermentable sugars. For this purpose, extracellular cellulase can be used. Bacteria like Cellulomonas species and fungi Trichodermata are good sources for cellulase. For the production of SCP from high energy sources, there are a large number of energy rich carbon compounds which serves as raw material for SCP production. Like these include alkanes, methane, methanol, ethanol, and gas oil. Bacteria and yeast are mostly employed for SCP from high energy sources. Now the last one is production from CO2. Certain algae grow in open ponds require only CO2 as the carbon source. In the presence of sunlight, they can effectively carry out photosynthesis and produce single cell protein. The examples of these algae are Chlorella species, Sandesmus species, and Spirulina species. After carrying out this, the product is further passed onto a ferment, then filtration and purification is done. After drying, the SCP is produced. If we look at the submerged fermentation, then it is the process in which substrate is always used in the liquid form, containing all the necessary nutrients needed for their growth. This whole process is carried out in a fermenter, which is operated in a continuous process, and the resultant biomass is harvested continuously using various techniques like centrifugation or filtration is done, followed by drying. The choice of technique used depend upon the microorganism, like bacteria can be recovered by centrifugation, while filamentous fungi require filtration for their recovery. The semi-solid fermentation, it is a process in which the substrate preparation is an important factor as it is used in solid state. This type of fermentation requires high capital investment and operating costs. So this is all about the industrial production of single cell protein. The single cell production in India, it is produced in India at NCBRI, that is National Botanical Research Institute. Here the SCP is produced on sewage, and at CFTRI, that is Central Food Technological Research Institute. In CFTRI, SCP is produced from algae culture on sewage. The application of SCP include it is used as health, food, and controls obesity, provide instant energy. The example include spirulina, and it is also used as protein supplemented food, which is also sources of vitamins, minerals, amino acid, fruit fibers, and it is supplemented food for undernourished children. These are the references from where I have taken the all information. Thanks for listening.
Very good, Anisha. So some question I have just sent in the chat box. Can you reply?
Yes, sir.
Anisha,
Yes, sir.
are you able to see some question in the chat box?
Yes, sir.
Yeah. You have given a very good presentation. The presentation was very good. Can you try to give this answer?
Yes, sir. Sir,
So
is streptomycin safe to use?
it's safe.
Yes.
Okay. What you can do, maybe you can just write it down this question that I have asked, and maybe you can write it down in page and submit in the Google Classroom. Can you do it?
Okay, sir.
Okay.
Yes, sir.
Then I can, because that is something like I want to see that how you have something like a new way of thinking, right? Are you
Okay,
getting?
sir. Yes, sir.
Yeah. So okay, all the best, Anisha. You can write down this question. I will create a space in the Google Classroom. And how much time do you think? Do you think you will submit by tomorrow?
Sir, I will submit by today only.
Okay, very good. All the best, Anisha.
Thank
So,
you, sir.
Nehal,
Yes, sir.
are you ready?
Yes, and I'm ready. Sir, actually, my laptop is having only 54% battery, and light is not there in my house, and inverter is also not working still, but I'll try to give faster.
Okay, you can give me 10 minutes.
Okay, sir. So please,
Okay.
okay, I'm going to share, sir.
Oh, please, please.
Is it visible, sir?
Yes, visible.
Okay, sir. Good morning to all of you. Today, I'm going to present my topic of enzymology, that is production of lysine and glutamic acid. Okay, basically, before moving to the further this topic into the deeper, that what is lysine and what is glutamic acid, how the production is used. First, let me know to all of you what are amino acids. See, basically, amino acids. These are biological important organic components contain amino group, that is NH2. We all know these, and carboxylic group, that is COOH, as a functional group. This is very basic part. We all know about that the amino acids are a biological organic components, usually with a side chain of specific to each amino acid. So the key elements of amino acids would be oxygen, hydrogen, carbon, nitrogen, though other elements are found in the side chains of such amino acids. Okay, so in the form of protein, such amino acid compromise, in the form of protein, I'm going to speak. You have to listen so carefully because I'm not including so much written in this presentation, but I'm going to present. So presentation is all about speaking. So in the form of protein, such amino acid comprise second largest components, means water is the largest of human muscle cells and other tissues. Outside protein, amino acids perform critical role in the process. Outside are like in the human body, water. We all know water is so much essential for our body. But outside the outside the tissues other proteins such as amino acids. These perform a critical role in the process, such as neurotransmitter, transport, and biosynthesis. Okay, let's talk about now production of amino acid first, that is lysine. I'm going to tell you production of two amino acids, that is lysine and glutamic acid. Okay, so in this case, in this class or in this presentation, I'll be discussing about the lysine and glutamic acid. First is lysine. See, lysine, it is an essential amino acid for the nutrition of human. As I told in the previous slide that amino acids are very much important. So lysine, it is an essential amino acid for the nutrition of humans. You know how it has been abbreviated lysine as LYS and capital K encoded by the codons like AAA and AAG. It is an amino acid used in biosynthesis of protein, like 2,6-diaminohexaonic acid is used. It is an alpha amino acid, and alpha carboxylic group also. And the side chain is lysine. Classifying it as an aliphatic amino acid. You know, lysine is an aliphatic amino acid and it is essential in the human. Means body cannot synthesize it has to be obtained from the diet. बॉडी अपने आप ये जो अमाइनो एसिड है लाइसिन, बॉडी अपने आप इसको सिंथेसाइज नहीं करती, इट हैज टू बी ऑब्टेन फ्रॉम द डाइट. सो it is an essential amino acid and it can be as a charge aliphatic amino acid and essential in human. It means body cannot synthesize, it has to be obtained from the diet. Okay. Supplementing cereal proteins lacking amino acids, it is used medically as a nutrient. And the lysine was just we all know it was discovered by the Kinoshita in the 1958 year. It was the first to report the fermentative production of lysine using homoserine auxotroph of this is the organism, uh, this cyanobacterium glutamicum. Okay, this cyanobacterium glutamicum. This is an important organism. Next, see, the features of lysine. Actually, let me not go to the deeper into fermentation process. Let me not go deeper into the fermentation process or the types of fermentation that are happening. Let us concentrate rather the media composition and the other conditions such as culture condition, operation mode, downstreaming process. As Anisha also in this previous slide, upstreaming and downstreaming process she discussed, and that every organism condition might differ. As you know, temperature, pH, we should having a little bit idea that what exactly the temperature is, what exactly the pH is. Means what are the exactly the requirement of this particular organism, of these particles having, like this medium composition, culture condition, operation mode, downstreaming process. These are the important features that are used in the formation of fermentation of lysine. So first is medium composition. Carbon source is required, that are being glucose, fructose, sucrose, and the soluble starch. And the nitrogen source being are ammonium, nitrate salts, yeast extract, soya bean protein, hydrolysis. Okay. And the culture condition, pH. pH would be 7 for this as it is neutralized by ammonia or urea. And the temperature would be 28 to 30 degrees centigrade. The temperature would be this. High oxygen concentration, it needs a more oxygen concentration. Means these are the features of lysine. You know how we are going to production of this lysine by fermentation process. These are the features, means these are the conditions that are required: medium, culture, operation mode, and downstreaming process. And it needs a high oxygen concentration. Operation mode, obviously, we have read that it is a stirred tank bioreactor, batch culture, fed culture, designed with the high oxygen transfer. Okay, and the downstreaming process. These downstreaming process also include the four major steps such as this cell separation. Means centrifuge or ultrafiltration can be used in the concentration like cation exchange resin and the elution with the alkali and neutralization with the hydrochloric acid and crystallization with the spray dried. Okay, lysine biosynthesis. See, two biosynthetic pathway exist for the amino acid lysine. First is DAP pathway, that is diamino pymalic acid. DAP. It is an intermediate that is found in bacteria, algae, and higher plants and oomycota. These are the lysine biosynthesis. This in the further next slide I'm going to explain with the diagram also, but these two biosynthetic pathway exist for the amino acid lysine. What first is DAP pathway, that is diamino pymalic acid. It is an intermediate that is found in bacteria, algae, higher plants and oomycota. Means pyruvate plus aspartate that is converted into DAP, that is diaminopymalic acid, and this is converted into lysine. So it is an important reaction, or we can say it is an important this pathway for the biosynthesis of lysine, that it is a DAP pathway. Second is AAA pathway. AAA pathway is alpha amino adiptate, means capital A. It can be abbreviated as capital A. It is an intermediate that is found in fungi and euglenoids. Okay, acetate plus alpha glutarate that is converted into AAA, that is alpha amino adiptate, and further it is converted into lysine. So we came to know that two important pathways are DAP and AAA. You know, animals do not synthesize lysine because it is an essential amino acid and animals do not synthesize it. So the production, this I'm going to read, that the chief ingredients in the medium were 2.5 glucose and 0.5 NH4Cl and the mineral salt, and the strain grew and accumulated L-lysine in the purely synthetic medium among the different hydrocarbon and nitrogen source tested. Because in the previous slide I told you many of the nitrogen source, oxygen source, and these are the concentration which are going to use, like glucose 2.5%, NH4 that is 0.5. Okay, gas oil this is 4%, and ammonium sulfate at 0.4%, that is respectively were found to be most suitable. Different vitamins and antibiotics stimulated growth and L-lysine yield. Inoculum of 7% of the medium was found to be optimal. The yield of L-lysine under optimal condition was 3.4 gram per liter medium. Actually, this image is not much clear. Sorry. See this glucose. This glucose is converted into aspartic acid. We know this Krebs cycle, we have already read. This glucose is converted into aspartic acid, and this aspartic acid in the presence of this aspartate kinase, it is converted into aspartase phosphate. This is converted into aspartase phosphate, and this aspartase phosphate is converted into aspartate semi-aldehyde. This is converted into aspartate semi-aldehyde. This arrow is here. This is converted into aspartate semi-aldehyde, and this aspartate semi-aldehyde is converted into dihydropimolic acid, and this dihydropimolic acid is further converted into diaminopimolic acid, and further last step is lysine. Then it is converted into lysine. So this was the biosynthesis of L-lysine in the Corynebacterium. This applications, the applications used, it helps to manage the cold sores causing by herpes simplex virus. It helps in the formation of bones and muscles, improving the athletic performance, boosting the immune system, and promoting insulin secretion. And last, I want to tell the production of glutamic acid. This is also an essential amino acid. We all know. Okay. Before explaining again, I want to tell you about the basic what is glutamic acid, as we discussed about what is lysine. The lysine was alpha amino acid and the alpha carboxylic group. And what is this glutamic acid? Glutamic acid is also an important amino acid, and it is abbreviated as G, L, U, or capital E encoded by the codons GAA and GAG. It is an alpha amino acid used in biosynthesis of protein. It is also an alpha amino acid and alpha amino group of C double bond O group. And alpha carboxylic group, we know that is the side chains are C double bond COO acid. Carboxylic acid means, and classifying it as a polar negative charge. This can be, glutamic acid can be classified as a polar negative charge. And aliphatic amino acid, this is an aliphatic amino acid, same. And see, it is not essential in human. Non-essential in humans means non-essential. What is the meaning of non-essential in humans? Means the body can synthesize this. Like in neuroscience, it is a carboxylate and glutamate. It is an important it's an important excretory neurotransmitter. Means here it plays a principal role in the neuroactivation. Like body this but
Nehal, Nehal, Nehal,
huh?
I think your voice is breaking, maybe network problem.
Audible.
Yeah, now okay.
Is it visible, sir?
Yes, yes.
Okay. Okay. I was telling about this glutamic acid. It is a derivative of glutamic acid, monosodium glutamate, and it is used to develop a flavor in food products. Hydrolysis of wheat, gluten, soya bean, cake,
And,
and
and
proteination.
Nehal, so Nehal, in fermentation, how do you produce this glutamic acid?
So this also include various steps like this. We need a ferment medium composition excrete this culture condition operation mode like batch culture, fed batch cultures, and by the downstreaming processes also. Answer, this was the biosynthesis of glutamic acid, which I have explained with the diagram, that how glucose is converted into glutamic acid.
Okay, very good. But you have explained very well.
Yes, sir. Okay, sir. I am going to only explain only with 2 minutes more that these are the important features of glutamic acid. Like same medium composition, excretion induction, culture, operation mode, downstreaming process. See, the sources being same: glucose, fructose, sucrose, and nitrogen sources being urea. And excretion induction means like biotin limited medium, addition of detergent, antibiotic, and pH would be 7 to 8, and temperature would be 30 to 34 degrees centigrade. Oxygen is required. Operation mode would be batch culture and fed batch. Downstreaming process, we know that these are the methods. Like so you were asking what are the methods, these are the methods like cell separation, concentration, crystallization. And MSG is a monosodium glutamic acid that is freely soluble in water. MSG means monosodium glutamic acid that is freely soluble in water, but it is not hygroscopic and is insoluble in the common organic solvent like such as ether. It is not soluble in the ether. It is generally stable under the food processing condition, and it has been used more than 100 years to the season food, and it was discovered and identified in 1866 by the German chemist Karl Heinrich Fritzen, who treated wheat gluten with the sulfuric acid. Means he was the first who treated this wheat gluten with the sulfuric acid. See, this is the basic step. Glucose is converted into acetate. Acetate is converted into acetyl coenzyme, then citrate, then isocitrate, then alpha ketoglutarate and alpha glutamate. In the presence of ammonia, NH4, it is converted into glutamate.
Nehal,
हाँ हाँ साक्षी
नेहल स्लाइड्स नोट स्लाइड्स विजिबल नहीं है
विजिबल नहीं हो रही शायद स्क्रीन मेरे ख्याल से लेफ्ट हो गई एक सेकंड
I am here. I am here.
अच्छा सर
Okay.
एक्चुअली हो सकता है स्क्रीन बस लास्ट हो गया सर ये ओनली
Okay.
इज इट
Okay.
विजिबल साक्षी
In between, it was not visible.
साक्षी सर इट इज विजिबल नाउ
Yeah, it's visible.
Okay, this was I was explaining this glucose is converted into acetate. Acetate is converted into acetyl coenzyme A. Then it is converted into citrate. Citrate is converted into isocitrate and then alpha ketoglutarate. In the presence of ammonia NH4, it is converted into glutamic acid. See, further steps are succinate, fumarate, malate, and oxaloacetate. Further, this is, but we were in the production of glutamic acid. This was the production. Applications important in metabolism, used in treating neuropathic diseases, and it is a flavor enhancer. Plant growth and is 30% oxygrow and various industrial application. Industrial applications such as thickener, bitterness, relieving agent, protectant, neurotransmitter, biological adhesive, and this pologlutamic acid is a naturally occurring biodegradable ionic polymer, and it is a heavy metal absorber. So this was all about my slide, that how the production of this amino acid, uh, two main amino acid, lysine and glutamic acid takes place.
So
Thank
you
you, sir.
so Nehal, Nehal.
Yes, sir.
So it was a very good presentation. I have asked some question in the chat box. Can you see it?
Yes, sir. I have clicked the photo of the same. I will submit in the evening.
Okay. One question that I'd like to ask.
Yes, sir.
When you were studying about lysine,
Yes, sir.
have you found something correlation of lysine for viral disease?
Sir, viral disease.
Yeah.
Sir, in the respiratory infection, it could be in the respiratory infection, like so, like food. In it could be like sir, as in the biosynthetic pathway, I said that AAA, that is alpha. This it cannot be stored in the human body, so it could get intestine diseases such as acid. It could be a viral disease, colds or infections.
Okay. Okay. Anything you have found?
Sir, disease. I'm not getting you. Sir, question like you are asking about yes, yes, it is good for viral disease, sir. Because lysine is when it is interfere with this arginine and it is being absorbed into our intestine, it makes the amino acid unavailable for the virus.
Very good. Okay. So now I have asked some question. That is one is the correlation of amino acid with immune response.
Okay, sir. Okay.
Okay. So that will give you the answer about other viral diseases, including COVID-19.
Okay, sir. I will try to find out.
Okay. And okay. So when do you think you will submit?
Sir, if today I'll complete in the evening, then today, or by tomorrow morning I'll submit before the class.
Okay. Okay, very good, Nehal. Can you just give me one thing? What does it mean by the functional group?
Like sir, side chains. As I told, this functional groups, these are having the side chains and
So suppose in an amino acid, we having the amino group and
yes,
we have the carboxyl group, right?
yes sir, amino group NH2 and carboxylic group that is COH.
Okay, so left side
It
is
is
the amino
it is
group and the
responsible
left side is the amino group and right side is carboxyl group, right?
carboxylic. Yes sir. I have
Now, now I have synthesized a new amino acid, and suppose now the carboxyl group is the left side and amino group is the right side. What will happen?
so what will happen?
नेहल आप बात समझ पा रही हैं क्या बोल रहा हूं मैं?
यस सर, वो जो NH2 राइट साइड पे है और कार्बोक्सिलिक लेफ्ट साइड पे है, अगर आप उनको रिवर्स कर दो, मतलब
हाँ हाँ
अपोजिट कर दो
हाँ
सर, तो आई डोंट थिंक कि रिएक्शन ऐसे कुछ हो पाएगी
तो वो सेम
सर। वो
रहेगा.
डेरिवेटिव हो जाएगा, मींस
वो अमीनो एसिड रह पाएगा कि दूसरा कुछ बन जाएगा?
नहीं सर, वो आई डोंट थिंक कि वो सर, वो जो रिएक्शन होगी बाद में बायोसिंथेसिस में, दैट वुड बी हेल्पिंग इन आफ्टर दिस इफ यू विल रिवर्स दीज टू ग्रुप्स।
Okay, so I have asked this question in the chat box. You can just write what do you think, and
यस
in the Google. You have done a good job. And then tomorrow we will start next presentation. So
सर,
have a
ओके
good
सर,
to all of you.
थैंक यू सर।
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.